Hello.
Hi.
Hi, how are you?
Okay, I guess. I don't know. No. Not busy anyway.
Oh, okay.
Well, I am because I have to catch up to things around here, but
Yeah. All right. How is everyone? Looks like we got a couple people. Mehul, welcome. Haven't seen you in a while.
Hi, Bradley.
Hi, how are you?
Hi, everyone. Hi, Susan. Hi, Dick. Hi, Soham.
hi. Hello.
Yeah, I hope everyone's doing well.
Yeah, just got my thesis published. It's actually online. You can download it for free for some reason. I just popped up my feeds and I went, is it for free?
Yeah.
And yeah, it downloads right away.
Oh, that's very nice.
Yes.
Yeah, so Bradley, actually Soham and I have been talking for some time on LinkedIn. We were discussing, although I couldn't contribute much to the discussion, Soham was asking a few questions. And I think, yes, last week he had a meeting also, so he briefed a little bit what was there in the meeting.
Okay.
Yeah. So I thought like I'll join. I also told Soham and also I discussed with you about the paper. So yeah.
Yeah.
Regarding that, yeah.
Okay, that sounds good. So you were talking on LinkedIn, you said, or okay, what were you talking what were you talking about?
So that mainly it was a discussion like how Soham can contribute. So like I have been pointing him towards the hypergraph part only, and I shared him the paper, your paper basically, which like the original hypergraph C elegans paper, and so Hum is trying to actually find like the downstream task, and that is what I was struggling also. So we were discussing a little bit on that. So Soham actually brought a good point, but the issue was there is not much papers he can find. So one of the application is of course cell tracking.
Okay.
And another thing which Soham mentioned was, I think, let me just check it. Soham, you can just unmute and tell.
Yeah, it was something Dick mentioned in the last meet about error accumulations in cell. So so if we could model that using hypergraphs, but I couldn't find much literature on that topic.
Yeah, I think that's something that we kind of were talking about in the meeting. I don't think there's going to be a lot of references on it, but yeah, that would be good. Any other points, or were those just the main two?
Yeah, that is it about discussions, and we thought we'll discuss more once we hop on a meeting.
All right, that sounds good.
Yeah.
So cell tracking, of course, is a part of the data sets that we get. So we were able to get data sets for different biological systems. We might take some microscopy of an embryo or something, and then we can track the cells basically using machine vision. We can figure out where the location of the cell. It's usually the cell centroid, or it's some sort of feature in the cell or cells, and you can use that to track things over time. So we could do something with tracking over time depending on the data set. The data set we usually use is uses like it doesn't really use nuclear cell tracking. It's sort of figuring out the centroid of the cell, and then that's what gets reported. But you can do this, sort of on board with a cell tracking package, or you can do this after the fact as well with like a machine learning algorithm. It's not, it's hard, but it's like, you can figure it out. And so that's kind of where that is. And, that's interesting for a number of reasons. We can track sort of the centroid of the cell. We can track maybe things like the nucleus or things like the membrane or other things in the cell that we might be interested in. And of course, since it's not just one cell, it's a population of cells, then we have all of those data. And tracking, especially with cell division, where you have one cell and then suddenly it's two cells, and you track that over time because you have time-lapse microscopy. You have this divergence of one cell into two or two cells into four, and that's something that you can put together. We've done that over several years, trying to work out different methods for doing it. So actually, if you go back to the DevoWorm repository, the DevoWorm software actually does a lot of stuff, interesting stuff with that, working with cell segmentation and cell tracking and trying to figure out how to put those things together.
Yeah, cell segmentation.
Yeah. Then other. Yeah, I'll go ahead.
No, please go ahead.
Yeah. So like with error accumulations in cells, the thing we were talking about last week, that's something that we can model. It wouldn't necessarily come from the data per se, but it would be like this. We know kind of what the cells, the cells are dividing, where they're dividing from, things like that, and you just you could build a computational model to pull out maybe a model of error accumulation, for example, and that's something we could work on, but that could be incorporated into a hypergraph model as well, I think. And it's just a matter of knowing in C. elegans we know the lineage tree, where we know if where a cell is in the embryo, if it divides, we know basically what the nomenclature, we know what the name of the cell is, we know what its properties are. Other embryos, we're not as lucky because the cells just kind of differentiate, in situ. They don't have a strict lineage relationship. So you see, that's why we work in C. elegans a lot of times. But this is something we can do as well. I would think you'd have a few other things we might incorporate into the model. And I don't know, maybe that's two things is enough. But I think the cell tracking is pretty much, a matter of going back to the data set. And, because we kind of train the model on a data set, and it's just kind of like we can do more with that. We can train it on other types of data. We can get other types of things in there. So that's something we can work out. I think it's more about playing with the data itself. With error accumulation, I think that's more of like we'd have to build a separate model of that and use that to, characterize each point in time, each set of cells that divide and things like that. Yeah, and I think that's probably, and I assume this is like a summer timeline, a GSOC timeline, so probably that would be enough for that, for like a summer project. There may be some other things that lead out of that, but I'm not sure what those are.
Yeah, that's very helpful. Thanks, Bradley. Definitely helpful for me and hopefully for Soham also.
Okay.
Yeah. Bradley, other thing which I wanted to discuss was about paper. So I really loved the paper, honestly. It was a great read. But one thing I felt was we haven't put a lot of empirical results. We did not had much also, but it will be good that if we can put a lot of metrics. Right now we are on the stationary part, so put a lot of hypergraph metrics in it, but also extend this extend this paper only, not create a new draft. Maybe also add dynamicity, since we already have C. elegans data. Putting the analysis of the dynamicity, how it is evolving over time. I think that will be pretty good, and it will make a more solid paper than it already is. So what I was thinking is this January and by the end of February, I'll try to complete as much analysis as I can, on the hypergraph, and show up in the meetings, discuss how should I which direction should I proceed, taking feedbacks and all. That is what I plan to do.
Okay, well, that sounds good. Yeah, I think that's true. We could use a lot of we could use a lot more information about how they unfold over time. So Dick says there is a literature on bilateral fluctuations during development. So yeah, if you have like an embryo and you have like, you establish poles in the embryo, and there are fluctuations in what happens in those two poles. Yeah, so let's see, I'll write that down and look into it after the meeting. That might be something to introduce as well. There are all sorts of like sort of meta properties to embryos that we could incorporate into this. And since this basically, once you get cell division, once you get polarity established, there are going to be a lot of things we can look at or consider, build into the model, etc. Yeah, so this is distinct from normal left-right asymmetry, like placement of heart. Yeah, so like in once you start to get like some of these left-right axes established, then you get things that develop asymmetrically, like the heart develops on one side of the body or another. And you don't see that as much in C. elegans, but that's something that there is that asymmetry. That's something. It's, I guess, kind of an open question in developmental biology, as I recall. Yeah, so that's something we could incorporate too. Yeah, so let's go over the paper. I've been working on revising it. So basically, this is the paper. They're talking about hypergraphs demonstrated in Anastomoses during divergent integration. And this, I put Mehul on here already, so the Soham wants to be on it as well. That's fine. We're just kind of I'm just trying to prepare a new version so that we can incorporate some of what Mehul did last summer. So this is the abstract. Basically, it's building these hypergraphs. So we have an earlier paper on embryo networks, which is where we take the cell tracking data that I talked about earlier and use those cells as the nodes in a network, and their edges, which are like things like distance, or they could be signaling, or other types of things that link together nodes. Basically, we have these hypergraphs, which are taking these nodes, putting them into larger categories, using what we call hypernodes to characterize groups of cells or groups of nodes, and then having these hyper edges, which are collections of edges. And we can do different things with this kind of model. So it's a higher level model than an embryo network. And so this paper kind of goes through a lot of what that is, using this analog of C. elegans to show kind of how as cells divide and differentiate, you get these different categories of cells, and that those categories sort of break out over development. You get exchange of cells between them, and it kind of establishes this. You can have like a directed graph, or you can have an undirected graph, or like I guess a semi-directed graph. So that's we do there. We also do something with a cyanobacterial species, Nostoc punctiforme, which is where we have this chain of cells that grows as the cells divide, and that's a different representation of that hypergraph method. And then we have this hyperdevograph model, which is incorporated in the last part, which is a computational approach. So there are there's a theoretical approach, there are two examples, and then there's a computational approach, which is the thing that Mehul worked on last summer. So this is setting up what we have here, and we go to the first graph. This is a category theoretic representation of exchange between the categories. And then we have this is the one I wanted to show. This is the directed hypergraph, where you start with a single cell. You get these, you get this sort of undifferentiated network. You start to get things like a germline, which is another hypernode, and then you get this embryo hypernode, this connectome hypernode, and you can see as the cells increase in their number through cell division and differentiation, you get these different hypernodes that represent different classes of cell. And so you have embryo cells, you have connectome cells, germline cells. In some cases, you get exchange between the two categories, between two hypernodes that involve like changing cells changing their fate, cells changing their category, but for the most part, you get a continuation of the categories as they grow. So that's what this is all about. And it's just like kind of a theoretical sketch of what that should look like. It's kind of tied loosely to C. elegans development, but we could easily build this with data that's available for C. elegans. This is a spatial hypergraph, so this is, I guess you could call it a temporal hypergraph. There's no spatial component. This is a spatial component where you have anterior-posterior organization and left-right organization of these hypernodes. The hypernodes then are connected over time to new hypernodes. So basically, this is a 128 cell example where you have 128 cells in these 4 hypernodes. And then later you get these new hypernodes that break apart. So if you go from 6, you end up with a doubling event, which is 12. Those are distributed in the following way. You get 2 cells in one hypernode here, 8 cells in another hypernode, and 2 cells that contribute to this other hypernode, which takes from another hypernode and combines things. So you can combine cells into new categories. You have doubling events, which increase the number of cells, and those get distributed into new hypernodes. And so that's what this is. This is a 256 cell example where you get 4 categories and of different types. You have the same 3 type, actually you get 4 types in this, but you get a greater number of hypernodes. Again, this is a theoretical example. It isn't really tied to the data so much, but it does give, it's loosely tied to the data just to give an idea of what's going on here. Okay, and then this is the example from Nostoc punctiforme, where you get this chain of cells. I think we talked about this in a meeting, and I kind of worked up this example. So we have this restricted heterocell differentiation along this chain of cells, and the idea is this chain of cells grows in length, and you can incorporate these cells into hypernodes. And as you continue on in time, this chain grows larger and you get these hypernodes that increase in size, but also you can get other types of hypernodes that branch off. I only show here these 3 examples. So it's restricted heterocell differentiation over time and applying this kind of method to this type of system. It's a simpler system than, say, something like C. elegans. And in turn, if you had something like a mammal or something like that, you would have an even more complex hypergraph representation than this. This is relatively simple. So just to give an idea of what we're expecting. Okay, so then I have some methods in here. Actually, this is taken from some data that is incorporated in the paper. These are some secondary data sets where, these are these are pseudo data where I kind of analyze these theoretical examples. And then we have actually some other data in here where we actually, provide some analysis. I can't remember where those graphs are right now, but anyways, there's some sort of pseudo data showing what the data look like. There's some actual data analysis showing what categories should look like in, say, like human cells and things like that. And then we have these graphs here. These are graphs from Mehul's work from last summer. So we actually implement a hypergraph using an algorithm, clustering algorithm. And so this is using the actual C. elegans data set this shows kind of how this was done, this clustering algorithm where there's this, these clusters are defined for different cell types. And we have these graphs here. And so, we have the regular lineage hypergraph, which is this first example A, and then we have the spatial hypergraphs for B and C. So the spatial hypergraphs are using this clustering as the connectivity criterion, whereas a lineage hypergraph uses common parents as the connectivity criterion. And so this is what we're, kind of trying to sort of replicate what we did in the theoretical examples, but using an algorithm on real data. And so it's harder to interpret what these are actually. And that's something, that I don't know how to approach in the paper. And maybe Mehul can help me with that. Like, how do you interpret this? But that's, if we get these different types of categories, we get these different types of connectivity criterion, and we can build then our graphs from that. Or, we can plot them out in more of a graphical form. So that's what we have now. Just kind of talking about future directions, and that's the paper. So, I think we've got a nice paper. I think, we need to have, maybe some, I think we could use some analysis, in different areas. I'm still working on it, so I was showing you work in progress there. But that's that we do have a preprint of this on the arXiv, but we're gonna be updating that soon, and then probably again. So, any questions about that?
Bradley, so like some thoughts, maybe like the point you mentioned, interpreting hypergraphs and that in more detail. I think what we can do is focusing on particular hypernodes and removing the other kind of cells. So in different snapshots, covering, having the coverage of different kind of hypernodes. So that can provide much clearer interpretation or understanding of these cells, maybe.
Yeah, I think so. Yeah, and then,
yeah.
Oh, go ahead.
Yeah, I had like some other questions also, but I think I'll give the draft like one more read specifically about the diagrams, like which you had restricted heterocell differentiation. But to be more precise, to have more precise questions, I'll go through them again.
Okay. Yeah, I mean, just collect your questions. I think that's more useful to kind of get a list. But yeah, I think that's good. I think that's good. I think that'll come out. I mean, basically the structure is we have sort of the theoretical aspect of it. We have some kind of thinking about what the data should look like. And then we have the computational work at the end. And I want to kind of do that sort of organizational scheme, because it's kind of establishing the problem, thinking about it in terms of actual biology, or at least kind of thinking about it in terms of the way we might see it in biology, and then, what we might expect to be able to use it for, some sort of spectral analysis, which is usually what hypergraphs can give you. They give you this sort of spectrum of information for different hypernodes, what are the constituents of the hypernodes, and being able to plot that out. And then the algorithmic work, which is, of course, implementing it on real data sets and, having it in these kind of, using different criteria to establish connectivity. Because, connectivity, it's almost talked about as this theoretical thing. It's like, we know things are connected. We want things to be connected. How do we establish that? In addition to, how do we gain how do we collect this from data where the data don't lend themselves to that type of organization necessarily? We have to impose it through some method. So that's I think that's the kind of organization we'll have. And then, of course, I think the algorithmic part might be, might benefit from some of these, visualizations where you're able to see kind of what the algorithm is doing, in terms of the cells, having those big visualizations, but also the smaller, maybe taking one hypernode and looking at what's in it, or, maybe looking at how you could build kind of, actually something that's connected. I don't know what kind of visualizations we could do, but we'll have to think about that. Because I took the images from like what you were doing last summer. So, I mean, I don't know, but
yeah, even like those images were like just came out of the blue. Like I wasn't thinking about producing those kind of images at that time.
Yeah.
Probably we'll discover on the way.
Well, I think it's good to have those, like, in terms of like understanding the basic types of connectivity, but then, having like a little bit more detailed, how do we get to, how are they interpretable? I guess in terms of biology is a thing. So yeah. So that's that's ongoing work. So Dick put in the chat some things on. This is on papers on fluctuating asymmetry. So I don't know how many papers are here. About 12 papers actually, and so there are a bunch of papers on this topic. There's this paper by Uchida et al. Deciphering the origin of developmental stability, the role of intracellular expression variability in evolutionary conservation. There's Danescu et al. Symmetry and fluctuation of cell movements in neural crest, derived facial mesenchyme. There's this other one. High temperature and oxygen supplementation can mitigate the effects of hypoxia on developmental stability of bilateral traits during incubation, boiler breeder eggs. There's one on zebrafish. There's one on Rutilis, delayed consequences of extremely low frequency magnetic fields and the influence of adverse environmental conditions on the roach, which is Rutilis. So there are a lot of different papers on that topic, basically looking at how these asymmetries get established in development. And, using these kind of representations are useful for uncovering the origins of that asymmetry. So thank you, Dick, for those references. We'll see, if that plays. I know we might put that into the paper as well, a section on developmental asymmetry or fluctuating asymmetries and things like that. All right, so let's move on. I have a paper I wanted to go over. I think this was shared in the OpenWorm Slack. So this is a paper on C. elegans, the modeling. So this is from Nature Computational Science. This is by a Chinese group. The title of this paper is Integrative Data-Driven Model Simulating C. elegans Brain, Body, and Environment Interactions. So this is, I don't think we've covered this work. I think we covered, it may be actually something that we covered when it was a preprint. I'm not really sure. It sounds familiar, but I'm not. Well, let's go over the paper to see what they're doing. So they're interested in not only modeling the worm, but modeling the body and environmental interactions. So again, they state that the behavior of an organism is influenced by the complex interplay between its brain, body, and environment. And if you look at C. elegans in its natural habitat, it usually exists in this three-dimensional habitat in soil. So it moves around soil, up and down different columns of soil, across soil. I mean, we're talking about an organism that's a few millimeters large, and it's navigating, I don't know what the range for a single C. elegans is, but where you can usually find C. elegans as in an environment where there's rotting fruit or some other type of, where you get things that fall on the ground and start to rot in the soil. And they like that kind of environment because it provides them with a lot of bacterial nutrients. And so I've actually harvested C. elegans from like a compost pile, which is interesting. And so you can do that, and they exist in this three-dimensional soil matrix. So, a lot of the things that the cybernetic group in OpenWorm has done is model this biophysical environment. They model the worm moving through the environment, and this environment is usually some sort of material. So they model it as like a material matrix where the worm moves through it. So, the matrix might be of different varying stiffness or varying, density, and the worm moves through that, and you can actually model that process. So the C. elegans environment is going to be, in a culture condition, you might have it on a two-dimensional plate where there's a gel and it's moving around the gel. And that's not really its normal mode of interaction. It's usually the soil environment. So it can vary, and you can have, but to model this environment is something that typically we often don't think about when we do a lot of computational studies on organisms. But anyways, existing data-driven models focus on either the brain or the body environment. Here we present BAAIWorm, or BAAIWorm, an integrative data-driven model of C. elegans, which consists of two submodels. So we have two submodels. We have the brain model and the body environment model. So we have these two sort of partitioned models of C. elegans. The brain model was built by a multi-compartment model approach with realistic morphology, connectome, and neural population dynamics based on experimental data. So this is kind of what we usually do when we model C. elegans. We have these different compartments. We have the morphology. We have the cells. This is the sort of thing that we're doing with our hypergraphs. We have the connectome, which are the neural cells, how they're connected functionally, and how they function. And then neural population dynamics, which are the dynamics of those cells in a connectome. So what are the cells doing to produce behavior? And of course, we can measure the activity of cells in the connectome and get these neural population dynamics. Okay, so that's the brain model, and we have these different compartments in the brain model that contribute to the overall this brain model. And then, of course, we also have a body environment model, which uses a lifelike body and a three-dimensional physical environment. So again, we have this three-dimensional physical environment where it's some sort of material where there are interactions with, not just on a flat surface, but in this three-dimensional volume. They're interacting with other worms, they're interacting with soil, they're interacting with other things in the environment, food, they're orienting themselves towards chemical signals, and so forth. Through the closed loop interaction between the two submodels, remember we have the sort of the brain and the body environment submodels. BAAIWorm reproduced the realistic zigzag movement towards attractors observed in C. elegans. So C. elegans will make this, zigzag movement towards different stimuli, and that's what they're trying to get at. So if you have like this food source, it's usually some sort of chemical source that they're trying to get to. They have these stereotypical movements they'll use to get to the source of the food, and that goes through this environment. So they're zigzagging through this volume of soil. So there's a specific resistance to the soil. It's a colloid, so it's moving in a certain way. It also has these stereotypical movements it's executing, which you can observe in culture. And you want to reproduce that. You also want to reproduce sort of this environment where you can train it on different, chemical stimuli and reproduce these chemical gradients that exist in nature. So they're able to set this model up, reproduce a lot of the natural behaviors we expect. And then leveraging this model, we investigated the impact of neural system structure on both neural activities and behaviors. Consequently, BAAIWorm can enhance our understanding of how the brain controls the body to interact with its surrounding environment. So this is what they're trying to get at. So this is where they talk about the different things that exist for this. They say currently there are specific data-driven models that simulate either the brain, such as the mouse primary visual cortex, the mouse striatum, and the rat somatosensory cortex, or exclusively the body and environment to reproduce animal behaviors, such as neuromechanical models for Drosophila and rodents. Then they talk about OpenWorm, where OpenWorm developed data-driven models for both the nervous system. This is the C302 model, and the body cybernetic. We talked about cybernetic of C. elegans, which is a pioneering initiative in computational biology. However, the integration of these models is open loop, meaning that they lack feedback from the environment. So again, this is, every time you build a biological model, oftentimes you make decisions on how you represent things. And sometimes it's easier not to include the environment explicitly. And, especially providing feedback to the actual worm. So in cybernetic, it doesn't provide feedback to the worm. It just kind of models the worm as this agent that's moving through a biophysically plausible environment. And it's just kind of figuring out how this biophysics work. In this case, they're actually including feedback. The same thing with C302. You have this connectome, you have the cells, you have the connections, you have the neural dynamics, and you have outputs, but oftentimes those are influenced by feedback from the environment. And so that's what they're trying to do here. So they say that basically their work fills a hole in the literature, which is that no data-driven model currently meets all requirements of an integrative data-driven model, although such integration is urgently needed to progress our understanding of neural control mechanisms. So yeah, there's been a lot of work on some of these. So let's see what they have. Reference 3, reference 4, 5, reference 6, 7, 8, 9, 10. And so let's go to those references, 3 through 10. Let me go down to the bottom. All right, here we go. So they're talking about these papers 3 through 10. We start with this paper, systematic integration of structural and functional data into multi-scale models of mouse primary visual cortex. This is a neuron from 2020. The second paper, the microcircuits of striatum and silico. This is from PNAS in 2020. You notice that these papers aren't that old. This is where a lot of the work's been getting done. This paper by Makram et al., Reconstruction and Simulation of Neocortical Microcircuitry. That's from Cell. This paper, NeuroMechFly, a neuromechanical model of adult Drosophila melanogaster. This is in Nature Methods, so we're going from mouse to Drosophila, so we have this variety of organisms. And usually you'll use these organisms because they're model organisms. We have data on them, and we've already kind of characterized the system of interest quite well. There's this other paper, Deep Neuroethology of a Virtual Rodent. This is a conference paper. Then we have the OpenWorm papers. We have this OpenWorm paper that's sort of the main paper that was published in 2018. We cover all aspects of the OpenWorm Foundation's project portfolio. We have this paper by Porrig Gleason et al., This is on C302. And then Andrei Polyanov and Sergei Karulin. And those are the guys who developed Cybernetic, a software complex based on the PCI SPH algorithm aimed at simulation problems in biomechanics. So there's this, this representative sampling of these different whole organism simulations. It's not just C. elegans, but they're adding into this milieu, I guess. Okay. So let's go to this. This is an overview of their model. They have a neural network, which is this sort of body. I guess it's just the brain submodule. This is where you have a neural network with neurons, which are multi-compartment models with real electrophysiological features. You have connections, which are detailed synapses and gap junctions on neurites. And then optimization. So you basically have the neurons, the connections, and the optimization. And that's their neural model. And then they have the body environment model, which is this down here where they model the body with tetrahedrons and they add muscles to those tetrahedrons. So they don't actually model it at the cell level per se. They're using these tetrahedrons to build a body, and then they're adding muscles to the body. So this is an interesting way to approach this because you're not really worried about the somatic cells so much. You're interested in the integrity of the worm and the shape of the morphology. So they use these tetrahedrons and then add muscles into them. Then you have the environment, which are lighting effects, visualization, tracking, quantification. This is not modeling it in a soil matrix. This is modeling it in this sort of environment that they've created for the, so that they have this, these sources of feedback. And then real-time simulation, which is where they have things in the environment that the worm has to interact with and gain feedback from. And hopefully, it incorporates into their behavior. So this is basically sensory muscle activation in the worm. You get the worm interacting with its environment. These blue dots, which are the sensory sources, you get the sensory activation, and it contributes back to the muscle activation. So in other words, the worm will move towards sources of sensory stimulation, and it will move its muscles as a result. So it's this closed loop feedback mechanism. So that's basically what they're trying to achieve. This is a biophysically detailed neural network model of C. elegans. This is where, of course, you get the morphology, the ion channel models, the electrophysiology, the connectome, the synapse and gap junction model, and then the network activity. Those are sort of the basic data collection components. Then you get this component construction where you get multi-compartment single neuron models and connections. You get model construction, which is where you have this biophysically detailed neural network. And then you have the model optimization, where you take the dynamics into account and you see how this neural network operates over time. So basically, you're looking at activity in the neural network and the connectome, whereas here you're just building the connectome from parts. And so this is a biomechanical body model. So notice now we're moving to the body. We have these this body that basically is this set of tetrahedron, and we have these serve as vertices for the sort of mesh model that they're using. So that's for the body. The actuator are these muscles. So the actuator is how this this geometric model moves. You have the solver, which projects dynamics into the future. So basically, as the worm moves, what should it look like? And we project that behavior outward. And we need to use solvers to do that because it's basically trying to solve for 96 muscles, 96 degrees of freedom. So this is pretty complex work. And then we have these, what we call the hydrodynamics, which are these sensory sources. You get thrust and drag of the body relative to the sensory sources in the environment. We can characterize that using hydrodynamic simulations. So again, this is something that, depends on the substrate, right? You can use hydrodynamics models and they characterize a substrate, to varying degrees of success. All we're interested in is the general relationships between the body and the environment. And so this is an internal view of the body model. And then this is the triangulated mesh. And then, using that as the body. Okay, so this is where they quantify the body's behavior. They have they're trying to calculate this trajectory of the body. So the body is here, and it's moving around in space, and you get these transformations. So the body's position back here is going to project to this point over here in the future, and as the worm moves and it kind of follows this trajectory, this line here is the trajectory of the target body center. So the target body is moving along this path, and you get this transformation that you have to project, which is basically when this part of the body moves along this trajectory, it moves to a different position. So as the worm moves and undulates, it has to predict the different positions on the body and how they transform in space. So this is what they're trying to do in terms of predicting or projecting out movement in the organism. This example in B is where you get this, you have this coordinate system of the standard body head, coordinate system of the target body head, and you basically track this over time. So this is, a tracking problem. C shows how the worm is kind of tracked in space. So as the worm is moving, it's a series of tracking trajectories, and that's basically how it reconstructs its position over time. So as it's moving, it's following these trajectories. Notice that there are multiple trajectories that are tracking different parts of the body, and then that's being extrapolated out into the future. And then you have the steering angle, which is calculated from sort of the angle of movement in the future that the worm experiences. So this is all kind of tracking motion in the environment. This is relative position of 3D tracking points. Again, we have position X, position Y, and position Z in time. And this is going from head to tail, so these different trajectories are from head to tail, top to bottom, or anterior to posterior. And it's these are the tracking positions here. So we can get relative position and relative velocity. So we have this velocity component, which is, of course, not just the position over time, but how it speeds up and slows down over time. So this is impressive work. They have this closed-looped interaction, as we mentioned before, where you have sensory activation, which feeds into the neural network model. Then it outputs muscle activation, which then affects the body environment model, which then affects sensory activation. So it's this closed-loop interaction, and they show the simulation and then they show experiments. So they actually have worms in a two-dimensional gel in culture where they're looking at the movement of the tail in this case. They're tracking the tail, and the tail does the zigzag as it's moving across the gel. And you have these different parts of the worm that are being modeled in the simulation, and this is what you're observing in the experiment. So it's able to reproduce what's going on in the experiment quite well. Of course, there's a lot more noise in the, I guess, well, I don't know what they're kind of extrapolating this line out, but yeah, I think it matches up somewhat. So it's basically producing the kind of output they want. And then they have these different things where they have in the simulation, they're measuring the motor neuron membrane potential, so they have the different motor neurons listed here. They're able to look at the membrane potentials and simulate those. And then the muscle cell activation. So in C. elegans, we have specific muscle cells, and we can look at their activation. So again, one advantage using C. elegans is we have specific cells that we can track by nomenclature and then produce an activation for each one. We know their position in the worm, so we can actually, account for this on a cell-by-cell basis and be very explicit in how these things happen. So this looks like where they're simulating the connectome, and they're actually doing some interesting things here where they're removing different parts of the connectome and they're getting different types of behaviors. So this is where they have like sort of a control connectome, and they have this sort of this connectivity map. I'm not sure exactly what this refers to, but basically it produces an output of locomotion. So for the control, it's producing this realistic type locomotion. You can see the trajectory here. You can see the worm. In B, what they're doing is they're removing neurites. They're getting a different, so this is a connectivity matrix here of principal components for the connectome. In the control, you see that the principal components matrix has a certain sort of shape to it. Then when we remove a neurite, or remove neurites, we get some change to this connectivity matrix, and we get a change to the output behavior. When we shuffle locations, which is, I guess, shuffling cells in the connectome, we get this connectivity matrix that's a little bit different than what we get in the control, but it still preserves some of the structure of the control, and we get this behavior that's modified. It's just looking at the trajectory, it's much more compact. The shuffling the synapse weights, we get a much different connectivity matrix, and we get this behavior where I don't know if it's maybe less robust. I guess it's kind of hard to interpret these behaviors, but you see changes in behavior as you do these different manipulations. You can shuffle the gap junction weights. Of gap junctions are the electrical connections between the cells. We usually have like a chemical connectome, which is where we have different synapses that we account for, or we have a gap junction connectome where we have different electrical connections between cells, or direct connections between cells that we account for. So we have two different types of connectome in C. elegans. In this case, we have a gap junction connectome, and we shuffle those gap junction weights so that we change the connectome in an appreciable way, and we get this output in this shifted output in behavior. We can also remove synapses in the chemical connectome, and we get this change in the connectivity matrix, and we get this change in behavior. We can also remove gap junctions and get something similar. So you see what they're doing here is they're building this principal component matrix of the connectome from like the data-driven connectomes that we have available. They make changes to those connectomes, and then they look at the locomotion output and see if it's this control or if it's changed by making these manipulations. And the reason we want to do this is because sometimes you get mutant phenotypes where there's a mutation in the genome, and those mutations sometimes can affect how the worm moves. And so you can get these mutations that affect the connectome and its structure, and it shows that those can affect movement. Sometimes those mutations are not viable. In other words, if the worm can't move, it's not a viable phenotype. But sometimes they are, and it just alters the movement patterns. So that's why they're doing this set of experiments. But actually, they have some more mathematical modeling down below, but I'm not going to get into that right now. But yeah, that's an interesting paper. And again, they're kind of going for this niche in the whole organism modeling area, which is to say that they have this they're trying to look at this brain-body environment set of interactions. And it's not as easy as you'd think to get those interactions. We kind of talked, I think we talked about, maybe a couple years ago now, where we tried to characterize environment. And what does it mean to have an environmental effect on something, especially in development or in behavior? When the environment changes or when the environment has an effect, what is that? What is the sort of signal that's being sent? What is the effect? Is it just something we say because it's, nice to say, and it just gives us a stand-in for something we don't understand, or what is it? So I think they kind of get at some of these things that are happening in the environment and how to incorporate them into a whole body model of behavior and of movement. So it looks like Dick had to go for another meeting, and Mehul had to go. So are there any other comments?
Looks like it's more complicated than what I put together.
Oh, well, I don't think you're trying to do the same thing. But
No, although it does look like it could be a tensegrity.
Yeah, you could probably build a tensegrity out of that.
And as I've found out, it's really hard to connect them. And actually, that's why they do single cells a lot. It's hard to connect tensegrity models together.
Yeah.
They don't seem to cooperate with each other.
When you say tensegrity models, you mean more than a few cells. So if you wanted to model
if you build a nice tensegrity model, it stands upright, you can manipulate it, and then you want to connect it to another one, the exact same thing.
Yeah.
Connect them together. Well, it doesn't work that well.
Oh.
It doesn't work to connect them together. Yeah, which is interesting.
Yeah.
Or a huge headache in my case. Yeah.
Yeah, I think that's true of most modeling paradigms, like where you try to connect things together, like from you have these multi-compartment models which allow you to sort of model things as compartments, but like a lot of times getting the dynamics to interact well. Like if I were to get connect like two connectomes or something, that would be kind of hard to do, or connect the effects between two connectomes or two worms. How could I do that without blowing the simulation up? And
yeah, well, in my case, it was my dog's playing with the squeaky toy.
Yeah.
I could only connect it with certain tri-junctions.
Right. Yeah.
Yeah. Anyways, yeah. Oh well, if you read my paper, you can see that I can only use the simple ones on the outside and the more complicated one in the middle. And,
Oh, okay.
That was one of the main things about that thesis. This is how I managed to connect these tensegrities together.
Yeah, I've been looking at your thesis, and it's interesting. I haven't gotten really deeply into it though.
Okay. Well, the first part of it is what is the physical model of a cell using cytoskeleton? Like, what is it?
Yeah.
And then the second part is to make something that works, that follows that.
Yeah.
I basically used the simplest tensegrities I could. If that's the simplest model I could come up with. So
yeah.
So it's a minimalist type tensegrity model.
Yeah. And then
Anyway, I would like to make a more complicated model, but yeah. I tried to actually. but then I couldn't get the my hexagons to line up.
Right.
Yeah, so I don't know what's up with that, but
Huh. Well, are you? Yeah, you're. I guess you're going to continue. We talked about this before. You're going to continue working on that.
yeah, I'll
Yeah.
continue working on that to make the 7-cell model. I'll try it, sure. And I want to investigate using other programs to make tensegrity models, but I'm not that interested in using command line code to make a physical model. I just don't want to. But like Linux, there's a program called Morpho, but it's Linux. So anyway, and it won't work on the computer that I'm on currently. I got a new computer. I'm going to try things out.
Yeah.
Yeah, so I'll keep working on things.
That's good. Yeah.
Oh well.
Yeah. Well, thanks. All right. Well, if there's no more questions or comments, let's call it a day. Thanks for attending, and see you next week. All right. Bye.
Bye.
Bye.